Good morning, my name is Massimo Bertoli and I am a PhD student from the University d'Annunzio of Chieti and the LETS laboratory of the Italian Research Council. First, I would like to thank the organizing committee for giving me the opportunity to contribute to this online conference with an oral presentation about the effects of a personalized neuromodulation against multiple sclerosis fatigue. We are interested in understanding and hopefully treating MS fatigue because it is one of the most frequent and bothersome symptoms of the disease, and at date there is no effective therapy against it, unless with the unpleasant side effects. We moved from the knowledge that MS fatigue origins from a functional rather than structural alteration, mainly involving the primary sensory motor and parietal networks, thus suggesting potential benefits from treatments that aim to compensate the neuronal electric communication dysfunctions. In particular, literature on MS fatigue indicates a specific involvement of the sensory motor network, with primary motor areas reported as excessively excitable, while S1 and primary and postparietal nodes of the somatosensory network less excitable than normal. In addition, connectivity between S1 and M1 appears to be impaired in fatigue patients and sensitive to alteration of neural networking. Based on this knowledge, our group carried out a personalized stimulation protocol called Faremos, that is fatigue relief in multiple sclerosis. Faremos consists in a 5-day anodal TDCS over bilateral S1 cortices in order to increase S1 excitability while trying to keep M1 unstimulated. Given the anatomical contiguity of these cortical areas, we used a personalized electrode shaped on MRI-derived individual cortical folding properly positioned over the target areas. The efficacy of this innovative approach for relieving fatigue in MS patients with minimal clinical severity has been observed previously by 2 independent RCTs, crossover and sham-controlled, that achieve an efficacy in relieving fatigue with a mean effect size of 1.3, that is a very large effect, as indicated by the fatigue levels reduction expressed by the modified fatigue impact scale percentage after the real treatment, 28% in the first group, 42% in the second. Moreover, LETS laboratory implemented a novel concept procedure for the identification of neuronal sources, called functional source separation, which proved capable of resolving cortical sources topographically close to each other, like M1 and S1. Here we applied the functional source separation algorithm to extract the activity of S1 and M1 hand representation in order to study their activity through time, that is, their neurodynamics, and the effects of Faremos on them. As you can see in this picture, we identified M1 activity of each hand during a motor task consisting in an isometric hand grip against a semi-compliant air bulb at 5% of maximum voluntarily contraction, maintained for periods of 20 seconds, here between the green and the red marks, intermingled by 10 seconds periods of rest to avoid task-induced fatigue. S1 activity instead was carried out from cortical responses to electrical stimulation delivered to the median nerve at the wrist of either left and right hand. Once disentangled the activity of M1 and S1, we were able to study their neurodynamics at rest. The experimental procedure included: at day 0 before the treatment, we performed the personalized electrode shaping based on individual MRI, once for each person with MS. In blocks 1 and 2, equal but each stimulation real or sham, we collected the data at rest, pre-Faremos, and positioned the electrode over the target areas for each TDCS stimulation, repeated for 5 days of treatment. After a period of time necessary to return to the baseline, in the 5th and last day we performed again the data collection at rest. The data collection, pre and post Faremos, included the fatigue levels assessment via modified fatigue impact scale and the EEG at rest for the brain effects. To assess the resting state local activity, we used the fractal dimension of the left and right primary sensory and motor cortices. We used the fractal dimension because it already proved to be a good candidate for assessing the brain network state and functionality, and also for typifying cortical districts at rest. Besides, we believe that complex system measures are proper to describe the physiology of neuronal electric activity whose dynamics display hugely complex temporal structures. To assess the resting state functional connectivity between the 4 nodes of the sensory motor network, we used the mutual information. Moving to results, we found that the Faremos neuromodulation had a distinct impact on the activity of S1 and M1, with major involvement of S1, in particular of the left hemisphere. In fact, as we can see here, pre-Faremos fractal dimension of the left hemispheric S1 turned out to be greater in fatigue MS patients than controls. Faremos mostly changed fractal dimension of left S1, and the difference seen before disappeared after the treatment. Of note, whereas S1 activity differed from healthy controls, this did not happen for M1. These results indicate a higher impairment of S1 with respect to M1, in line with our hypothesis that the parietal cortex mechanisms play a crucial role in generating MS fatigue, with the local impairment more evident in the dominant control region. Concerning intra- and interhemispheric functional connectivity between S1 and M1 areas, we observed that overall the mutual information values improved, mostly between the left and right M1. Pre-treatment, comparing the functional connectivity of healthy controls and MS fatigue patients, we can see that these latter had lower values. Post-treatment, the mutual information value improved significantly, reaching a level comparable of that of the healthy controls. Thus, rebalancing the sensory-motor functional connectivity results in a reinforcement of the interplay between homologous M1 areas that contributed significantly to fatigue amelioration. So, what did we learn about MS fatigue? The parietal, more than frontal cortical networks are involved with MS fatigue. Faremos neuromodulation rebalanced the sensory-motor functional connectivity, reinforcing the interplay between homologous M1 areas. The neurodynamics changed by Faremos, that is the fractal dimension and mutual information, explain together 48% of fatigue levels amelioration. Lastly, the fractal dimension among the complexity measures can quantify relevant properties at rest of the cerebral networking organization at the single node level. Thank you for your attention.